Hey everyone, in this lesson we are going to learn about gluconeogenesis. So what is gluconeogenesis? So if we break down this word, gluco means glucose, neo means new, and genesis means synthesis. So gluconeogenesis means basically synthesis of a new glucose from non-carbohydrate precursors. Now, why is gluconeogenesis important? In mammals, some tissues depend almost completely on glucose for their metabolic energy. And some of these tissues are brain, erythrocytes, testes, renal medulla, and embryonic tissues. And therefore, gluconeogenesis is very important for these tissues. Now, here are some facts. Did you know that human body requires 160 grams of glucose per day? And not only that, human brain alone consumes 120 grams of glucose per day, which accounts for almost 75% of glucose consumption. Now, glucose generated from the glycogen stores are equal to 190 grams, and glucose in the body fuel accounts for 20 grams. Now, gluconeogenesis is important during long intervals between meals, overnight sleep, longer fast, and after vigorous exercise, because these are the different situations when blood glucose level can go down. So basically, gluconeogenesis is actually important to maintain blood glucose levels during these different conditions. Now, gluconeogenesis primarily takes place in the liver, which accounts for almost like 90%, and to a lesser extent in the kidney and small intestine. Now, one important thing to remember is that gluconeogenesis is not merely the reverse of glycolysis. So in glycolysis, one glucose molecule is broken down into two molecules of pyruvate, and there are three different enzymes which play a very important role, and these are the three irreversible reactions of glycolysis. Whereas on the other hand, in gluconeogenesis, two molecules of pyruvate are used to synthesize one molecule of glucose. And most importantly, these three irreversible reactions of glycolysis are bypassed in gluconeogenesis. And these reactions are bypassed with the help of four different enzymes. So the first two enzymes, that is pyruvate carboxylase and phosphoenolpyruvate carboxykinase, these enzymes are important to convert pyruvate into phosphoenolpyruvate. Second is fructose 1,6-bisphosphatase enzyme, which converts fructose 1,6-bisphosphate to fructose 6-phosphate. And lastly, glucose 6-phosphatase, which is important to convert glucose 6-phosphate into glucose. So in gluconeogenesis, there are these four different enzymes which bypass the irreversible reactions of glycolysis, and these enzymes are very important. So first thing we are going to learn is how pyruvate is converted into phosphoenolpyruvate. So there are two steps of how pyruvate is converted into phosphoenolpyruvate. First step involves the carboxylation of pyruvate to oxaloacetate, and these reactions require bicarbonate, ATP, pyruvate carboxylase enzyme, and biotin. And the reaction also releases ADP and inorganic phosphate and two hydrogen atoms. Now, in second step, oxaloacetate is then converted into phosphoenolpyruvate with the help of enzyme phosphoenolpyruvate carboxykinase, and also known as PEP-CK. So now, I first want to talk about a little bit in detail how pyruvate is carboxylated into oxaloacetate. So for this reaction, first step is the activation of carbon dioxide. And for activation of carbon dioxide, there is an interaction between ATP and bicarbonate to synthesize carbonyl phosphate. And this is actually the activated form of carbon dioxide, and it also releases ADP. So carbon dioxide is linked to the phosphate group. Now, second step is the transfer of this carbon dioxide to biotin prosthetic group by the enzyme pyruvate carboxylase. And before we go into how the carbon dioxide is transferred to biotin, I first want to quickly just go through the structure of biotin and how biotin is linked to the pyruvate carboxylase enzyme. So here you can see that this is biotin and this is pyruvate carboxylase enzyme, and this is the lysine residue of the pyruvate carboxylase enzyme. So biotin is covalently linked to lysine residue of the pyruvate carboxylase enzyme. And here I just wanted to show you how biotin is covalently linked to the pyruvate carboxylase enzyme. So here is the simpler version. This is biotin, and which is covalently linked to the pyruvate carboxylase enzyme. And here we have ATP and bicarbonate. When they interact, they form this carbonyl phosphate, which is actually the activated form of carbon dioxide. So when carbonyl phosphate interacts with biotin, which is covalently linked to pyruvate carboxylase enzyme, they form N-carboxybiotin. So N-carboxybiotin is the complex of pyruvate carboxylase, biotin, and carbon dioxide. So the carbon dioxide from carbonyl phosphate is transferred onto biotin, which is linked to pyruvate carboxylase. And the reaction also releases inorganic phosphate. Now N-carboxybiotin then interacts with pyruvate to form oxaloacetate, and this third step involves the abstraction of proton from C3 of pyruvate. So when this proton is abstracted, carbon dioxide from N-carboxybiotin is then transferred onto C3 of pyruvate to synthesize oxaloacetate. And it also releases this biotin, which is covalently linked to pyruvate carboxylase enzyme. And lastly, there is an enzyme called biotinidase which cleaves off biotin from the active site of pyruvate carboxylase enzyme, and that way it releases this biotin, and which is then available to be reused for the next set of reaction. One important thing to remember about this reaction is the role of biotin, which is to carry carbon dioxide from carbonyl phosphate to pyruvate so that it can synthesize oxaloacetate. So this is the first step where pyruvate is carboxylated to form oxaloacetate. Now we need to learn how oxaloacetate is converted into phosphoenolpyruvate. So here it is, and I'm going to go through the carboxylation of pyruvate reaction again. So here we have pyruvate, which is synthesized from the breakdown of one glucose molecule, and that is through glycolysis, and we know that glycolysis takes place in the cytosol. Now this pyruvate is transported into the mitochondrial matrix via mitochondrial pyruvate carrier, and once inside the mitochondrial matrix, pyruvate is carboxylated to synthesize oxaloacetate, which requires this bicarbonate biotin ATP as well as pyruvate carboxylase enzyme. Now, oxaloacetate produced in this reaction cannot actually cross the mitochondrial membrane. Therefore, oxaloacetate must be first converted into malate with the help of mitochondrial malate dehydrogenase enzyme. And in this reaction, NADH is oxidized to NAD+, and malate, which is synthesized through this reaction, is then transported into cytosol through malate aspartate shuttle, and malate in the cytosol then converted back into oxaloacetate with the help of cytosolic form of malate dehydrogenase enzyme. And in the reaction, NAD+ is reduced to 2-NADH, and NADH then enters into electron transport chain to synthesize ATP. Now oxaloacetate from here then converted into phosphoenolpyruvate with the help of phosphoenolpyruvate carboxykinase or PEP-CK enzyme, and it also requires 2-GTP and releases carbon dioxide. So this is the two steps, where first step is the carboxylation of pyruvate to oxaloacetate, and second is the how oxaloacetate is converted into phosphoenolpyruvate. So just to give you a quick recap, we just learned that pyruvate is first carboxylated to oxaloacetate, and then oxaloacetate is converted into malate in the mitochondria with the help of mitochondrial malate dehydrogenase enzyme, and then malate then transported into cytosol, where malate is then converted back to oxaloacetate with the cytosolic form of malate dehydrogenase enzyme, and then oxaloacetate is converted into phosphoenolpyruvate with the help of phosphoenolpyruvate carboxykinase enzyme. Now the reaction of carboxylation of pyruvate to oxaloacetate consumes 2 molecules of ATP, and 2 molecules of GTP are also used when oxaloacetate is converted into phosphoenolpyruvate. Now phosphoenolpyruvate, through series of reactions, it is then converted into fructose 1,6-bisphosphate, and in between of this reaction, 2 molecules of ATP are being used when 3-bisphosphoglycerate is converted into 1,3-bisphosphoglycerate, and fructose 1,6-bisphosphate is then converted into fructose 6-phosphate with the help of fructose 1,6-bisphosphatase enzyme, which also releases one inorganic phosphate, and fructose 6-phosphate is then converted into glucose 6-phosphate, and glucose 6-phosphate is then converted into glucose with the help of glucose 6-phosphatase enzyme, and also releases one inorganic phosphate. So if we do the math, glycolysis synthesizes 2 molecules of ATP, whereas gluconeogenesis consumes 6 molecules of ATP. So gluconeogenesis is very expensive process, but it is worth it because many important tissues which just relies completely on glucose for their metabolic need, and we just learn about that in the introductory slide, and most importantly the brain. Now the other thing I want to talk about is how free glucose is generated from glucose 6-phosphate in the liver. So far we saw that fructose 1,6-bisphosphate is converted into fructose 6-phosphate with the help of fructose 1,6-bisphosphate enzyme, and fructose 6-phosphate is then converted into glucose 6-phosphate with the help of phosphoglucose isomerase enzyme. Now in most tissues, once glucose 6-phosphate is generated, gluconeogenesis stops here. It doesn't actually releases free glucose until it is required. In that case, glucose 6-phosphate is then converted into glycogen to store glucose in the form of glycogen. Now consider a situation when blood glucose level is low, then free glucose will be synthesized from glucose 6-phosphate. However, free glucose is not generated in the cytosol. Rather, glucose 6-phosphate is first transported through T1 transporter into the lumen of endoplasmic reticulum. And once inside the endoplasmic reticulum, glucose 6-phosphate is hydrolyzed to inorganic phosphate and glucose with the help of ER membrane bound glucose 6-phosphatase enzyme. And this inorganic phosphate and glucose, they are then shuttled back with the help of transporter 2 and transporter 3 into the cytosol. This is something I thought is very important to know, how free glucose is generated from glucose 6-phosphate. Now another important thing to know about is that liver and kidney are the only organs that release free glucose. This only takes place in tissues whose metabolic duty is to maintain blood glucose level. And liver is predominantly involved in maintaining blood glucose level, and then kidney. Now, I also want to talk about substrates which are required for gluconeogenesis or gluconeogenic precursors. So first is lactate. Through Cori cycle and anaerobic metabolism, it is actually an important precursor for gluconeogenesis. And I talked about Cori cycle in detail in my previous video, so please go and review the detailed mechanism of Cori cycle and how lactate is involved in gluconeogenesis. Second is fat, which involves glycerol, which is synthesized from the breakdown of triglycerides, and propionate from the degradation of odd chain fatty acids. Third is amino acids, and mostly all 20 amino acids are gluconeogenic precursors except for 2, which are leucine and lysine, and we will see shortly why they are not the precursor for gluconeogenesis. And lastly, monosaccharides such as fructose and galactose. So this is the pathway of gluconeogenesis, and here we are going to see how different precursors enter into the different stages of gluconeogenesis pathway to synthesize glucose. So here we have triacylglycerol or triglycerides in the adipose tissue. So in adipose tissue, it is broken down into fatty acids and glycerol. Now glycerol, through blood, it is then transported into the liver, and in the liver, glycerol is converted to glycerol 3-phosphate with the help of glycerol kinase enzyme, and glycerol 3-phosphate is then converted into dihydroxyacetone phosphate with the help of glycerol 3-phosphate dehydrogenase. And dihydroxyacetone phosphate is an intermediate of glycolysis. Now one thing I want to mention, that adipose tissue, they actually lack glycerol kinase enzyme, and because of that, adipose tissue cannot convert glucose into glycerol 3-phosphate, and that is actually the reason that once glycerol is synthesized in the adipose tissue, it is then transported through blood into the liver, and in the liver it can then be used to synthesize glucose. Now second is propionate, which is synthesized from the breakdown of odd chain fatty acids, and propionate is then converted into succinyl-CoA, and succinyl-CoA is an intermediate of TCA cycle, and succinyl-CoA is then converted into malate, and malate to oxaloacetate and to glucose. So propionate is another precursor for gluconeogenesis. Second is lactate from Cori cycle, which is converted to pyruvate with the help of lactate dehydrogenase enzyme. And again, if you want to review this in detail, please watch my previous video on Cori cycle. And third, glycogen, which is actually the storage form of glucose, through glycogenolysis is converted into glucose 1-phosphate and then to glucose 6-phosphate. And galactose, which is also then converted into glucose 1-phosphate to glucose 6-phosphate and then to glucose. And lastly, fructose, which is converted to fructose 1-phosphate and fructose 1-phosphate to dihydroxyacetone phosphate and then to glucose. So these are basically the precursors for gluconeogenesis. And now I want to quickly talk about amino acids, and these are actually called glucogenic amino acids. So glucogenic amino acids can be degraded either directly into pyruvate, or they can also be degraded into one of the intermediates in the TCA cycle, such as alpha ketoglutarate to succinyl-CoA to fumarate and to oxaloacetate. And all these intermediates can then be converted to oxaloacetate, which is actually the main precursor for gluconeogenesis, because oxaloacetate from here can then be converted into phosphoenolpyruvate and through series of reactions into glucose. Now these amino acids are named glucogenic because they can produce glucose under conditions of low glucose, and because they can be used in synthesis of glucose, that is actually the reason they are called glucogenic amino acids. Or other substrate, they can also be called as glucogenic substrate. Now I also want to talk about substrates which cannot be used as precursors for gluconeogenesis, and one of them is acetyl-CoA. Now in mammals cannot use acetyl-CoA as a precursor for glucose. And why? Because acetyl-CoA is synthesized from pyruvate through an irreversible reaction with the help of pyruvate dehydrogenase complex enzyme. And cells have no other pathway to convert acetyl-CoA to pyruvate. In fact, plants, yeast, and many bacteria, they do have a pathway where acetyl-CoA can be converted into pyruvate, which is known as glyoxylate cycle. And if you want, I can make a separate video on glyoxylate cycle. But what you need to remember that in mammals, acetyl-CoA cannot be converted back into pyruvate because this is an irreversible reaction. And from here, acetyl-CoA is then converted into acetoacetyl-CoA. And most importantly, as I mentioned, that out of 20 amino acids, there are 2 amino acids which are actually not the precursor for gluconeogenesis, and one of them is leucine, because when leucine is broken down, the final end product of breakdown of leucine is acetyl-CoA, and we know that acetyl-CoA cannot be actually converted back to pyruvate. And second is lysine, and lysine is converted into acetoacetyl-CoA and which is then converted into acetyl-CoA. So because both leucine and lysine, they have to go through acetyl-CoA, and acetyl-CoA cannot be used as a precursor for gluconeogenesis. Therefore, leucine and lysine also cannot be used as a precursor for gluconeogenesis. And also when triglyceride is broken down, it is broken down into fatty acids, and the catabolism of most fatty acids yields only acetyl-CoA, and therefore many of the fatty acids cannot be the precursor for gluconeogenesis. And also we learned that triglycerides can also be broken down into glycerol, and glycerol is an important precursor for gluconeogenesis. So this is an overview of precursors for gluconeogenesis, what are different kinds of precursors, and how they enter into the pathway of gluconeogenesis to synthesize glucose. And lastly, I quickly wanted to talk about the comparison of the pathways, which is gluconeogenesis and glycolysis. So in gluconeogenesis, 2 molecules of pyruvate synthesizes 1 molecule of glucose, whereas in glycolysis, 1 molecule of glucose is broken down into 2 molecules of pyruvate. Now, gluconeogenesis primarily takes place in the liver, which accounts for 90% of the pathway, whereas to a lesser extent in the kidney, as well as also in the small intestine. Now, glycolysis, which occurs in erythrocytes, cells of brain, kidney, and exercising muscle. It also takes place in other tissues, but these are the tissues which solely rely on glucose. Now, gluconeogenesis takes place in both cytosol and in the mitochondria, whereas glycolysis takes place only in the cytosol. There are 4 bypass enzymes of gluconeogenesis, which include pyruvate carboxylase, phosphoenolpyruvate carboxykinase, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. And 3 important enzymes for glycolysis include hexokinase or glucokinase, phosphofructokinase 1, and pyruvate kinase. And lastly, gluconeogenesis uses or consumes 4 ATP, 2 GTP, which are then hydrolyzed to generate 1 molecule of glucose. So basically, it consumes 6 molecules of ATP. Whereas glycolysis, it uses 2 molecules of ATP in the beginning, and then it synthesizes 4 molecules of ATP and if you do a math, it actually synthesizes 2 molecules of ATP from the breakdown of 1 molecule of glucose. And lastly, I also wanted to talk about the clinical significance of the enzymes involved in gluconeogenesis pathway. Now, first is biotin and biotinidase deficiency. So biotinidase deficiency is basically an autosomal recessive disorder, whereas biotin is actually nutritional deficiency. So biotin nutritional deficiency is actually caused by a large amount of avidin protein which is found in the raw egg whites, and this avidin from raw egg whites then binds to the biotin and makes it unavailable for attachment to the pyruvate carboxylase enzyme. So if the biotin is not able to attach to the pyruvate carboxylase enzyme, that means pyruvate cannot be carboxylated to oxaloacetate. And there is another one which is called biotinidase enzyme. And if you remember that the important function of biotinidase enzyme is to cleave off the biotin from pyruvate carboxylase enzyme. So it releases the free biotin to be used for the next cycle of reaction. And because this enzyme is deficient or this enzyme is mutated, it cannot release free biotin, and therefore it cannot be used to carboxylate pyruvate to oxaloacetate. Now, symptoms of biotin deficiency involve rashes, brittle hair, hair loss, anemia, dermatitis, fungal infection. So there are all other list of symptoms which can be caused because of the deficiency in biotin. Now, second is PEP-CK deficiency or phosphoenolpyruvate carboxykinase deficiency. It is an early onset autosomal recessive disorder which is caused by a mutation in PEP-CK1 or 2, and this deficiency, PEP-CK deficiency, causes defective gluconeogenesis, hypoglycemia, loss of muscle tone, and liver enlargement. Now, third is fructose 1,6 bisphosphatase deficiency, which develops in individuals with mutations that make this rate limiting enzyme defective. It presents in early childhood with hypoglycemia, lactic acidosis, ketosis, apnea, which is abnormal pauses in breathing, and hyperventilation triggered by fasting or ingestion of fructose, glycerol, or sorbitol. And last is glucose 6-phosphatase deficiency, which is actually a type 1A glycogen storage disease due to a deficiency in glucose 6-phosphatase, and it is characterized by severe fasting hypoglycemia because free glucose is not unable to be produced from either gluconeogenesis or glycogenolysis. So thank you so much for watching this video, guys, and I really hope that you learned something new from this video. If you do so, please like and share the video and subscribe the channel. And thank you so much, and I'll see you soon.